Imagine for a second that you are a liver transplant surgeon. You've just finished this grueling, life-saving procedure on a patient who has hepatocellular carcinoma, or HCC.
Right, which is the most common type of liver cancer out there.
Exactly. And replacing the entire organ is often, it's viewed as the ultimate miracle treatment. You did it. You literally removed the disease from the body.
You'd think so, yeah.
But, and this is the terrifying part, as you monitor this patient over the next few years, there is this massive, dark statistical cloud hanging over that miracle. In up to 40% of post-transplant cases, the cancer returns within 5 years. And if the tumor was just surgically resected, that recurrence rate jumps to a staggering 70%.
It is honestly one of the most devastating clinical realities in oncology today.
Yeah.
The patient goes through this massive, incredibly invasive procedure. They're convinced they have a new lease on life. And yet somehow microscopic fragments of that tumor manage to survive. They hide and they just come roaring back.
Which is exactly why we need to talk about how they hide. Today's deep dive is, well, it's pretty much required listening if you have any interest in cancer biology or immunology. We are digging into the source material of this groundbreaking 2026 paper published in the journal Immunity by Gu and colleagues.
Yeah, this paper's huge.
It really is. We're going to look under the hood to understand exactly how these recurrent liver tumors survive our immune system and, critically, how scientists have finally found a way to strip away their armor.
And this is where the paradigm completely shifts, right? Because the key to this specific cancer survival isn't a traditional immune shield. Like, it's not a protein that simply repels immune cells. It's a metabolic shield.
A metabolic shield.
Right. It all comes down to how the tumor manages its own internal supply chain of cholesterol. And we're going to unpack every single molecular mechanism of this today, step by step.
Okay, so as we walk through this, I want you, the listener, to keep a central question in the back of your mind today. If a tumor is utilizing its own unique metabolic diet to hide from our immune system, what happens when we forcefully change that diet?
That's the million-dollar question.
So let's start at the beginning. If you're a researcher looking for a hidden metabolic shield, where do you even begin to look?
Well, you start right at the bedside with the patients. The researchers anchored this entire study by looking at 28 human HCC liver transplant patients.
Okay.
And all of these parents shared similar clinical backgrounds, similar treatments, but they had divergent outcomes. 15 of them experienced a recurrence of their cancer, and 13 did not.
So you essentially have the perfect natural experiment right there. You've got a split group, which allows you to compare the molecular fingerprints of the tumors that came back versus the ones that didn't.
Precisely. And to figure out what the recurrent tumors had in common, they used this highly advanced analytical technique called liquid chromatography mass spectrometry, or LCMS.
LCMS.
Right. Yeah, and if you aren't familiar with it, you can think of LCMS as an incredibly precise molecular weigh station. It separates the complex mixture of molecules in the tissue and then measures their mass and charge to identify exactly what is there. So they performed proteomic profiling on the tumor samples, essentially taking a full inventory of every protein present.
Wow. And what did they find?
Out of all that data, they identified 86 differentially expressed proteins, or DEPs, between the two groups.
Okay, meaning there were 86 proteins acting noticeably different in the aggressive returning cancers. But, in biology, you're usually looking for the ringleader. Did one of those 86 stand out?
Oh, one absolutely dominated the data. It's an enzyme called SOAT1.
SOAT1.
Right, which stands for sterile teloacyltransferase 1.
Okay, if SOAT1 is our primary suspect here, what is its actual D job inside a normal cell?
So SOAT1 resides in the endoplasmic reticulum, or the ER. That's basically the cell's manufacturing and packaging center. Its specific job is to catalyze a very critical reaction. It takes free cholesterol, which, believe it or not, is actually quite toxic to the cell's membranes if it builds up.
Wait, really? Toxic?
Yeah, highly toxic in its free form. So SO1 converts that free cholesterol into stable cholesterol esters, or CE. Once converted, the cell can safely pack those esters away into storage units called lipid droplets.
Okay, let me offer a visual for that, because understanding this mechanism is really the bedrock for the rest of our discussion. Think of free cholesterol floating around the cell like highly volatile, loose gunpowder.
That's a good way to look at it.
Right, because if it accumulates, it's corrosive and dangerous to the cellular machinery.
Yeah.
So SO1 is the specialized packaging plant. Its entire job is to take that volatile gunpowder and safely seal it into secure, chemically inert wooden crates. Those are the cholesterol esters. And if you want a mnemonic to remember its function, Just think of the letters in est. Securely organizes and tucks away cholesterol.
I like that. Securely organizes and tucks away. It captures exactly what it does. And the clinical correlation they found in the source material is just stark. The researchers saw that patients who had high levels of this SOP1 packaging plant in their tumors had significantly faster recurrence times and much worse overall survival.
But wait, if they have higher levels of this enzyme, does that mean the tumors are just like obsessively hoarding these stable wooden crates of cholesterol?
Yes, exactly. When they looked at the actual tissue from the recurrent cases, they found significantly elevated levels of stored cholesterol esters. The tumor is essentially running its packaging plant in overdrive to stockpile fuel and protect itself from toxic buildup.
Wow.
And this doesn't just impact surgical outcomes, it drastically impacts immunotherapy. Patients with high SO1 had a much higher rate of progressive worsening disease when they were treated with immune checkpoint inhibitors like anti-PD-1 therapy. The tumors just brush the drugs off.
Okay, let's pause and make sure we are locking this foundation in before we go deeper. Based on the proteomics of these 28 transplant patients, we know high levels of SO1 drive liver cancer recurrence.
Right.
We know SO1's job is to protect the cell by converting toxic-free cholesterol into safe, stored cholesterol esters. And we know that tumors doing this are practically immune to our best immunotherapy drugs.
Correct.
Which naturally brings us to the intervention. And this is my question for you, the listener. If SO1 is the packaging plant, keeping the tumor safe from all this volatile cholesterol, what happens to the cancer cell if we permanently shut that plant down?
Well, the researchers asked that exact same question, and they attacked it from two different angles. First, they genetically deleted the SOT1 gene using CRISPR, creating what we call SOT1 knockout cells.
So basically completely ripping out the blueprint for the enzyme.
Exactly. And second, for a more clinical approach, they used a pharmacological drug called evasion, which is a specific inhibitor that binds to the SOT1 enzyme and just blocks its activity.
Okay, so the packaging plant is officially locked down. No more wooden crates being built. What happens to all that volatile free cholesterol?
It initiates a massive cellular catastrophe. Let's trace the mechanistic steps here. Step 1. Without SO1, free cholesterol rapidly and aggressively accumulates in the endoplasmic reticulum.
The gunpowder just piles up.
Right. And the researchers didn't just assume this happened, they proved it visually. They used a fluorescent stain called Philippin III.
Philippin III.
Yeah. What's fascinating about Philippin III is that it specifically binds to unesterified free cholesterol and glows under a microscope. So when they applied it and did targeted ER lipidomics, they could physically see the ER of these tumor cells just overflowing with glowing, toxic-free cholesterol.
And in biology, I mean, a drastic change in one area almost always sets off alarms in another.
Oh, always. Which brings us to step 2. This cholesterol buildup in the ER triggers a chain reaction involving two crucial proteins called SREBP1 and SRBP2.
Okay, SREBPs, what are those?
These are master transcription factors. Their job is to read the cell's DNA and turn on the specific genes that manufacture new lipids and new cholesterol. You can think of them as the cell's supply chain managers.
Okay, so if the cell is suddenly choked with all this free cholesterol, I'd assume those supply chain managers would realize they have way too much inventory and just shut down production, right?
They do, but the mechanism of how they do it is the actual trap we are exploiting here. Normally, when a cell needs more fat, these SREBPs travel from the ER into the nucleus to flip the on switch for production genes.
Right.
But the cell uses a chaperone protein called SK to sense cholesterol levels. When the ER is overflowing with free cholesterol, that cholesterol physically binds to SK.
Oh, I see where this is going.
Yeah, this binding acts like a cellular glue. It physically anchors SK, and the SREBPs attach to it firmly inside the ER. It's SK-mediated retention. They are physically trapped and cannot travel to the nucleus.
So it's a structural mechanism, not just a signaling one.
Exactly.
But the researchers must have had a way to verify that the supply chain managers were actually stuck, right?
They did. They used immunofluorescence to tag the SREBP2 proteins, and it showed a complete absence of them in the cell's nucleus. Because they can't reach the nucleus, we hit step 3. The tumor cell abruptly and completely stops synthesizing its own endogenous cholesterol and fatty acids.
Wait, hold on. This is where I have to marvel at the biological paradox here. By stopping the tumor from storing cholesterol, we inadvertently trigger a feedback loop, a physical trap, that stops the tumor from creating any new fats or cholesterol entirely.
That is exactly what happened.
So the tumor is drowning in a toxic buildup of free cholesterol, while simultaneously starving to death for specific newly synthesized fats.
It puts the internal environment of the tumor cell into a state of total metabolic panic, which we call ER stress. Because the ER is so compromised by all this, normal protein folding begins to fail. The researchers measured elevated levels of specific stress markers like IRO1, BP, and PDI. You can think of these proteins as the cellular 911 dispatchers. They are part of the unfolded protein response, and their presence in such high numbers proves the tumor cell is structurally just freaking out. It is starving for specific newly made lipids.
Let me summarize this metabolic cascade so we don't lose the thread here. So, bullet 1. By inhibiting SO1, we cause free cholesterol to pile up in the ER. Bullet 2. That buildup acts like glue, trapping the SREBP supply chain managers. Bullet 3. Because they are trapped, they can't turn on production genes, so the cell stops making new fats, resulting in massive ER stress.
Perfect summary.
But here's the bridge we need to cross, and I'll pose this to the listener. We've fundamentally broken the tumor's supply chain of new fats. But how does this internal metabolic starvation translate to the tumor suddenly becoming vulnerable to an attack by our immune system?
To understand that vulnerability, we have to look at the exact chemical fallout of that SREBP trap. Because the tumor can no longer make new lipids, it loses several very specific vital chemicals.
Like what?
Well, it loses unsaturated fatty acids. It loses metabolic intermediates like 7-alpha-hydroxycholesterol or 7-HC. But most crucially, its production of a molecule called prostaglandin E2 or PGE2 absolutely plummets.
PGE2, why is that specific molecule so important to a liver tumor?
Because PGE2 is a highly immunosuppressive molecule. Tumors aggressively synthesize it and pump it out into their microenvironment. Its primary function for the cancer is to basically sedate attacking immune cells, specifically CD8 plus T cells. It paralyzes their ability to fight.
So going back to our visual analogies, PGE2 is functioning like a chemical smokescreen. The tumor is constantly pumping out this thick smoke, and the T cells wander in, get blinded, start coughing, and are completely unable to mount an attack.
Yes.
But by deleting SO1, we broke the supply chain, the PGE2 production crashed, and the smokescreen cleared. The T cells can finally see the target.
Precisely. But clearing the smokescreen is only half the battle here. There's a second, equally lethal blow happening simultaneously.
Okay, what is it?
The lack of newly synthesized lipids absolutely destroys the tumor's internal antioxidant capacity. The researchers proved this by measuring something called the GSH to GSSG ratio, which drastically dropped.
Let's unpack that ratio really quickly. What are GSH and GSSG?
GSH is reduced glutathione, which is essentially the cell's primary sponge for soaking up dangerous oxidative damage. When it soaks up that damage, it oxidizes and becomes GSSG. A healthy cell has a high ratio of the protective GSH. But in these metabolically starved tumors, that ratio fell off a cliff. The sponge was gone. They had no way to neutralize oxidative stress.
So we didn't just clear the smokescreen. By stripping away those antioxidants, we essentially removed the tumor's internal fire retardant.
And that completely sets the stage for the kill. When CD8 plus T cells attack a tumor, they don't just, physically punch holes in it. They release a powerful cytokine, a weaponized protein called interferon gamma, or IFN.
Right.
Normally, a robust, healthy tumor just brushes interferon gamma off. It uses its vast supply of antioxidants to neutralize the threat and repair the damage.
But because we trapped the supply chain managers, the tumor has no fire retardant left to neutralize it.
Exactly. Without its antioxidant shield, the interferon gamma from the T cells triggers massive, unchecked oxidative stress inside the tumor cell. Specifically, it causes a phenomenon known as lipid ROS, or reactive oxygen species accumulation.
Lipid ROS.
Yeah. The vital lipids that make up the cell membrane literally begin to oxidize. The membrane degrades, it loses its integrity, and the cell undergoes a catastrophic structural failure. It fundamentally rusts from the inside out and shatters.
That is brutal, but incredibly elegant. And researchers, being researchers, I assume they didn't just guess that this loss of antioxidants was the exact cause of death, right?
Oh, no, they proved it definitively. They took these metabolically starved tumors in a petri dish actively being destroyed by T cells, and they manually intervened by adding a potent synthetic antioxidant called NSC, or N-acetylcysteine, back into the cellular environment.
Wait, they manually handed the tumor its fire retardant back?
They did. And the moment they did, the tumors survived the T cell attack once again. The lipid ROS accumulation stopped. It completely reversed the vulnerability, proving without a doubt that the loss of that antioxidant shield was the exact mechanism of death.
Okay, let's tie this section together before we move on. So first bullet, by trapping the SREBPs, We halt the tumor's production of both the immunosuppressive PGE2 and its vital cellular antioxidants.
Yeah.
Second, without the PGE2 smokescreen, the attacking T cells wake up and swarm the tumor. And third, without the antioxidant fire retardant, the T cells' weapons cause lethal lipid ROS to accumulate, shattering the tumor.
That's the mechanism in a nutshell.
Now, I have to play devil's advocate here and pose this to you, the listener. We have beautifully dismantled this cancer's defenses in a highly controlled lab setting, essentially in a plastic dish. But how on earth does this hold up in the messy, complex reality of a living body, especially a body that might be dealing with other massive metabolic issues like obesity or complex medications?
That is the ultimate test. And to answer it, we have to look at the broader clinical picture of liver cancer. Real-world hepatocellular carcinoma doesn't happen in a vacuum. It is heavily driven by metabolic disorders, particularly non-alcoholic fatty liver disease, or NFLD, and obesity.
Right, because obesity fundamentally alters the metabolic landscape and the lipid availability of the entire body.
Exactly. And the clinical data from the source material confirms this connection. They found that obese HCC patients defined clinically as having a BMI over 28 had significantly higher SO1 expression in their tumors. And consequently, they had significantly worse survival rates compared to non-obese patients.
So the tumor just thrives in that high-fat environment.
It loves it. So if the researchers wanted to prove this works in vivo, they had to recreate that hostile, fat-rich environment.
How do they do that?
They designed a study using mice, feeding them a high-fat, high-cholesterol diet, or HFHCD, for 2 solid months. This induced systemic obesity and fatty liver disease before they even transplanted the liver tumors into the mice. They were asking, can SO1 inhibition still starve the tumor if the entire circulatory system is practically flooded with excess dietary fat?
Talk about the ultimate stress test and the results.
Astounding, honestly. They treated these obese mice with a combination of SO1 deletion and anti-PD-1 immunotherapy. Now remember, earlier we noted that anti-PD-1 normally struggles against these SO1-heavy tumors.
Right, the progressive disease.
But here, the combination resulted in near-complete tumor eradication. When the pathologist looked at the tissue under the microscope, they saw massive swarms of CD8 plus T cells completely infiltrating the tumors.
So even in a system absolutely flooded with dietary fat, sabotaging the tumor's internal packaging plant was enough to clear the smokescreen and let the immune system win.
It was.
But there is one more massive real-world hurdle we haven't touched on. In fact, it's a paradox unique to liver cancer and transplantation.
Ah, you're referring to the immunosuppression catch-22?
Exactly. To cure this liver cancer, you often have to give the patient a brand new liver. But to stop the patient's immune system from rejecting that new foreign organ as an invader, doctors have to prescribe powerful immunosuppressant drugs for the rest of the patient's life.
Right, drugs like tacrolimus. Tacrolimus is incredible at protecting the new liver, but its mechanism of action is to literally paralyze T cells.
So if there is even one microscopic cancer cell left over from the surgery, you've just given it a free pass to multiply because you've medically turned off the very immune cells that would normally hunt it down.
It seems like an impossible situation, right?
Yeah. How did the researchers solve it?
By engineering an absolute marvel of cellular technology. They took CAR T cells, which are a patient's own T cells that have been genetically modified with specialized receptors to hunt specific cancer antigens.
Okay.
And they made them immune to the immunosuppressant drug.
Wait, what? How do you make an immune cell immune to an immunosuppressant?
By changing the lock so the drug's key no longer fits? Yeah. Tacrolimus works by binding to a very specific protein inside the T cell called calcineurin A, which shuts down the cell's activation. The researchers used gene editing to give their CAR T cells a mutant calcineurin A gene.
A mutant gene.
Specifically, they altered 2 specific amino acids, creating mutations known as T351E and L354A. These microscopic structural changes shifted the shape of the calcineurin protein just enough that the tacrolimus drug simply bounced off. It could no longer bind.
That's brilliant.
They created a tacrolimus-resistant CAR T cell, or TR-CAR T.
So you've engineered a precision biological assassin that completely ignores the patient's anti-rejection medication. It stays awake while the rest of the immune system sleeps.
Exactly. And when they deployed these specialized TR-CAR T cells into mice, combined with the SO81 inhibitor of avastin to strip the tumor's metabolic armor, the results were incredible.
I can imagine.
The immune cells aggressively hunted down and destroyed the liver cancer, all while the mouse was successfully flooded with tacrolimus, completely preventing the rejection of the transplanted organ.
They managed to thread the needle perfectly. curing the cancer without losing the transplant. Let's recap this final crucial leap into the real world. Bullet 1. So8-1 inhibition synergizes brilliantly with anti-PD-1 immunotherapy to eradicate tumors, even in highly obese models where the body is flooded with cholesterol.
Right.
Bullet 2. The inhibitor drug, ivospemin, preferentially starves the tumor without damaging the T cell's ability to fight. And bullet 3. By combining this metabolic sabotage with mutant TRCAR T cells, doctors have a viable blueprint to eradicate returning cancer without triggering the rejection of a life-saving transplant.
Which really brings us to the profound paradigm shift this paper represents for the future of oncology. I mean, for a century, we have largely thought about cancer therapy as a search for the perfect poison.
Yeah, chemotherapy, radiation.
Exactly. The foundation goal has always been to find a blunt force instrument that kills the tumor just slightly faster than it kills the patient.
It's historically been a game of cellular attrition.
But this research shows a completely different, much more elegant future. We don't necessarily have to poison the tumor. We can simply tweak its metabolism.
Tweak the supply chain.
Right. By disrupting its internal lipid supply chain, we make the cancer cell so fundamentally brittle, so incapable of handling basic oxidative stress, that our own natural immune system can easily sweep in and finish the job.
It's biological jujitsu.
Yeah.
You are using the tumor's own insatiable greed for cholesterol against it. And that brings me to a final provocative thought I want you, the listener, to mull over today as we wrap up. So8-1 is just one single enzyme. It manages one specific type of fat and one specific type of cancer. But the human body contains literally thousands of these complex metabolic pathways and molecular packaging plants.
Oh, the metabolic network is unimaginably vast.
Exactly. So if simply disrupting how a liver tumor stores cholesterol is enough to cure treatment-resistant cancer in these advanced models, what other metabolic vulnerabilities are hiding in plain sight? What other deadly diseases are relying on supply chains we just haven't learned to break yet?
It's a huge question.
Could it be that cellular metabolism, the very way our bodies process our diet and our lipids, holds the ultimate master keys to immunity? It fundamentally changes how you look at a tumor on a scan. It's not just a physical rogue mass to be cut out. It's a living, breathing metabolic network that we are finally learning how to unplug.
That's exactly what it is.
Well, thank you for joining us on this deep dive into the source material. Keep asking questions, keep challenging your assumptions about how the human body works, and we will catch you on the next deep dive.